closely related to our daily lives because when we are going to study around us, we see that there are so many things which is very much concerned to our daily life. So before going further, I would like to discuss some of the safety rules in the laboratory. So we can divide them into two safety rules. One is called as general safety rules. They do not enter general safety rules, and then we have different safety rules when we are going to mix some chemical or we are handling the chemicals. Sometimes we are heating some objects, so we have to face these things. And sometimes, unfortunately, we have some accidents. So what we do? So we'll discuss over here. First, we will talk about the general safety rules. So most important, listen to or read instruction carefully before attempting to do anything. Wear safety goggles to protect your eyes from chemicals, heated materials, or things that might be able to shatter. Notify your teacher if any spill to accident occur. Then, general safety rules: after handling chemicals, always wash your hands with soap and water. During lab work, keep your hands away from your face. General safety rules: roll up loose sleeves. Know the location of the fire extinguisher, fire blanket, eye wash station, and first aid kit. Keep your work area untethered. Take to the lab station only what is necessary. It is suggested that you wear glasses rather than contact lenses. Never put anything into your mouth during a lab experiment. Clean up your lab areas at the conclusion of the laboratory period. Never horse around or play practical jokes in the laboratory. Clipped or cracked glassware should not be used. Show it to the teacher. Broken glassware should not be disposed of in classroom trash can. There is a special glass disposal container for it. When pouring liquids into glassware, make sure that the container you are pouring into is resting on a table at least hands width away from the edge. Glassware safety: do not place hot glassware in water. Rapid cooling may take it shatter. Chemical safety: wear protective goggles and a lab apron whenever heating or pouring hazardous chemical. When mix chemical together, unless you are told to do so, and then only in manner or specified. Never taste any chemical. You should never taste anything in the lab. Pour down a glass tiring rod of preventing liquid from splattering. If a piece of glassware gets broken, do not try to clean it up by yourself. Notify the teacher. When inserting glass tube into a rubber stopper, apply a lubricant like glycerin to the glass and use a twisting motion. Chemical safety: if you need to smell the odor of a chemical, waft the fume towards your nose with one hand. Do not put your nose over the container and inhale the fume. Never pour water into a concentrated acid. Acid should be poured slowly into water. Chemical safety: follow the instruction of your teacher. It's very important when disposing of all chemicals. Wash your hands after handling hazardous chemical. Electrical safety: unplug cord by pulling the plug and not the cord. Unplug all electrical equipments at the end of the lab periods. Heating safety: left burner and hot places cool down before touching them. Test to see if they are cool enough by burning the back of your hand close to them. Use tongs and or protective gloves to handle hot objects. Never reach across an open flame or burner. The only type of glassware that may safely be heated in either Pyrex or Pyrex. Always point to the top end of the test tube that are being heated away from people. When heating a test tube, move it around slowly over the flame to distribute the heat evenly. Only glassware that is thoroughly dry should be heated. Heat glassware by placing it on a wire gauze platform on a ring stand. Do not hold it in your hand. When lighting a burner, wait until the sticker is in place before you turn on the gas. The amount of air can be adjusted by the air supply valve below the tube of the burner. This regulates the flame, temperature, and color. Never leave a burner or hot plate unattended. First aid, injury, burns. What to do? Immediately flush with cold water until burning sensation is lessened. First aid, injury, cuts, bruises. What to do? Do not touch an open wound without safety gloves. Pressing directly on minor cut will stop bleeding in a few minutes. Apply cold compress to bruises to reduce swelling. First aid, injury, fainting. Do not provide fresh air and have the person recline so that their hands is lower than the rest of their body. First aid, injury, eyes. What to do? Flush eyes immediately with plenty of water for several minutes if a foreign object is caught in the eyes. Do not allow the eye to rub. First aid, injury, poisoning. What to do? Find out what substance was responsible for the poisoning and alert the teacher immediately. Okay, dear students, as we have discussed the general safety rules of the laboratory, now you will see some of the apparatus. Laboratory apparatus, test tube, beaker, measuring cylinder, pipette, filter funnel, Benson burner, tripod stand, flask. Then we have evaporating dish, wire gauze, thermometer, rod stand and clamp, burette, test tube rack, stand, stopwatch, pistol and mortar. Then we have cotton stopper, spatula, glass rod, dropper, filter paper, test tube holder, crucible with lid and crucible top. Now here we have some of the steps which is used in scientific investigation. Number one, identifying a problem, formulating a hypothesis or forming a hypothesis, planning in the experiment, controlling variables, carrying out investigation, reporting. Identifying problem arises from a desire to know what happens in our surrounding. Example, why do birds fly but not hostage? Why do cactus able to grow on a desert land with no water available? Then we have forming a hypothesis. A hypothesis is an early idea or explanation for the phenomena that we observe. A hypothesis can only be acceptable or proven true after going through experiment. If the hypothesis is not true, a new hypothesis has to be formed. Planning an experiment. A list of material and apparatus required and the way they should be carried out are made before an experiment can begin. Moreover, we need to determine the way to collect, analyze, and interpret the data obtaining from the experiment. Controlling the variables. Variables are situations and factors that are controlled and changed in any investigation. Types of variable: a variable that is kept constant, variable that are kept constant or fixed, a variable that is manipulated change, variables that are used to test the hypothesis, a variable that responds, results are collected from the variable that are changed when testing a hypothesis. Carrying out investigation, steps that are to be followed. Collecting data. All data are observed and measured accurately. Number two, analyzing and interpreting data. The data is then presented in a table or chart. Interpretation is made from the table or chart. Making conclusion, an accurate statement to describe the problem identified throughout the experiment. Determine whether to accept or reject the hypothesis. Reporting. A report is made after conducting a scientific experiment based on the format shown. The title, aim, hypothesis, variable, A, constant. B, manipulated. C, respond. Material operators, procedure, result, observation, discussion, and conclusion. So these are the 4 steps what we discuss over here. Inshallah, in upcoming slide, we'll do some more thing regarding this chapter. Thank you very much. Jazakallah for today. Assalamu alaikum wa rahmatullahi wa barakatuh.